When most of us think about the domain eukarya, the group that contains all complex life, including us, we immediately picture the big three.
Right. Plants, animals, or fungi.
Exactly. But if you actually look at the family tree, those multicellular groups are just these tiny, specialized branches.
They really are. The overwhelming majority of eukaryotic diversity, both in terms of, sheer number of species and structural variation, is hidden in what we once just called the kingdom Protista.
So to be clear, what exactly is a protist?
It's basically a catch-all. A protist is any eukaryote that is not a plant, an animal, or a fungus.
And while, okay, most of them are single-celled, the idea that they're simple is just fundamentally wrong.
It's the complete opposite of simple. Because they don't have tissues or organs like multicellular life, a single cell has to do everything.
Everything.
Every single life, function, movement, feeding, waste disposal, reproduction, it all has to happen within its cellular boundaries. This forces them to be the most structurally complex cells in existence.
That level of subcellular engineering must be just astounding. Can you give us an example of what that complexity looks like?
Certainly. I mean, you see familiar things like nuclei and mitochondria, but protists also evolve these highly specialized organelles. For instance, some aquatic protists have contractile vacuoles.
And what do they do?
They're like tiny active pumps that regulate water balance to stop the cell from bursting. But even more bizarrely, some dinoflagellates have an ocelloid.
An ocelot.
It's a complex eye-like organelle. It has components that perfectly resemble a lens and a retina, all built from internal membranes.
A microscopic, single-celled eye. Wow. Okay, that really sets the tone for our deep dive today. We are going to map this staggering diversity, their structures, their functions, and especially their evolutionary roots, which really tell the story of how eukaryotes came to be.
And to really get it, we have to look at their flexibility. Nutritionally, they span the whole spectrum. You've got your classic photoautotrophs making food from light, your heterotrophs that consume organic matter, and then the really fascinating ones, the mixotrophs.
The ultimate survivors.
They absolutely are. Mixotrophs can switch their strategy. If it's sunny, they photosynthesize. If it's dark or there's food around, they just become heterotrophs and start eating things.
And I'm guessing their reproduction is just as varied?
Oh, yeah. Sexual, asexual, and they even display these really sophisticated life cycles we usually associate with plants, like alternation of generations. It's an evolutionary proving ground where, all the possibilities are explored.
To understand that incredible complexity, we have to go right back to the beginning of the eukaryotic domain. It all comes down to a few key moments of evolutionary collaboration, what we call endosymbiosis.
This is the foundation. The very first core event gave us the mitochondria. An ancestral host, probably an archaean or a close relative, engulfed an aerobic alpha proteobacterium.
So the host basically got this powerful little energy factory that could use oxygen, and the bacterium got a safe place to live.
Exactly. And the proof is right there in the DNA. Mitochondrial DNA confirms this link. What's so crucial to remember is that this was a single event, a one-time thing.
One time for all of us.
Yes. Every single eukaryote, from a tiny protist to a blue whale, is descended from that single common ancestor that first got the mitochondria.
That sets the metabolic stage. But what about photosynthesis? Where did that come from?
That came later in a separate event called primary endosymbiosis. A lineage of these new heterotrophic eukaryotes went and engulfed a photosynthetic cyanobacterium.
And that's where plastids came from.
That's it. That acquisition of plastids gave rise to the entire Archaeplastida supergroup, which include red and green algae and eventually land plants.
But the map gets messy here because so many other protists have plastids, but they are not in the Archaeplastida group.
And that's where the next layer comes in. Secondary endosymbiosis. This is where a heterotrophic protist didn't just engulf a bacterium, it engulfed an alga eukaryotic cell that already had a plastid inside it.
So it's literally that Russian doll of cells you mentioned, a protist swallowing an already photosynthetic protist.
Precisely. And this leaves a calling card. Plastids from primary endosymbiosis have two membranes, but those from secondary endosymbiosis, they usually have 3 or 4 membranes, which reflects the multiple layers of the cell that was engulfed.
That is just tangible evidence of these stacked evolutionary partnerships. It's wild that so much of life is built on these ancient events of one cell eating another and just deciding to keep it. Okay, now that we have their origin story, let's start wading into the classification, starting with the first major supergroup, Excavata.
Excavata is, it's really diverse. The name originally came from an excavated feeding groove some members have, but what's really unifying is how many of them evolved in anaerobic environments, which led to some extreme changes to their mitochondria.
Let's start with the diplomonads. They live where there's no oxygen.
Correct. They have no plastids, and their mitochondria are these highly reduced structures we call mitosomes. They can't use oxygen at all, so they rely on anaerobic pathways. The most notorious example is Giardia intestinalis.
Ah, the parasite that causes that awful persistent diarrhea.
That's the one. And a similar strategy is used by the parabasalids.
Right, they also have reduced mitochondria.
Yes, but theirs are called hydrogenosomes. Like mitosomes, they work anaerobically, but they release hydrogen gas as a byproduct. This group includes Trichomonas vaginalis, a very common human parasite.
So it seems like a lot of the protists that affect human health evolved these specialized ways to live without oxygen.
That's a very common theme. Moving on with an excavata, we get to the euglenozoans. They're defined by a unique structure inside their flagella.
What is it?
It's a crystalline rod that runs right through the flagellum. This group includes the plastids, which get their name from their single enormous mitochondrion containing a massive clump of DNA called a kinetoplast.
And the most famous example here, the one causing sleeping sickness, trypanosoma, has this insane defense mechanism against our immune system.
Oh, it's one of the most successful evasion tactics known. Imagine your immune system finally figures out what a surface protein on trypanosoma looks like and starts building an army against it.
Okay.
Well, before those defenses can even get going, the parasite just changes its entire protein coat to something brand new. It's a bait-and-switch defense.
It's constantly creating a new disguise, so the host can never develop a lasting immunity. That's genius in a terrifying way.
It is. And that's in sharp contrast to the euglenids in the same group, which are often just free-living mixotrophs, happily switching between photosynthesis and eating, guided by an internal light detector.
Okay, let's transition to the second massive supergroup, Sarchanzar. This is probably the most diverse and ecologically dominant group we'll cover. And it's named just by combining the initials of its three clades, right?
That's right, straminophiles, alveolates, and rosarians. The grouping is based purely on overwhelming genomic evidence.
Let's start with the Straminophiles.
They get their name from their distinctive flagella. They typically have a large, hairy flagellum covered in these fine hair-like projections, and that's paired with a short, smooth one.
This group includes some global powerhouses, like the diatoms.
Diatoms are just phenomenal. They're unicellular algae encased in these unique glass-like walls made of silicon dioxide, which fit together like a tiny box and lid. But their structure isn't even the main story. It's their ecological role.
Right, they're huge global producers. But what does that mean in terms of scale?
Diatoms and other photosynthetic protists account for about 30% of the world's photosynthesis. They're the base of the entire aquatic food web. And crucially, when they die, their heavy glass shells make them sink fast.
Taking all their carbon with them.
Exactly. It transports atmospheric carbon locked in their bodies straight to the ocean floor. They're essentially a massive biological carbon pump.
Absolutely essential. Also in the Straminophiles are the largest protists of all, the brown algae. We know them as multicellular seaweeds like kelp.
Yeah, and they display a complexity that makes them look almost identical to plants, with specialized structures for anchoring and photosynthesis, and they perfectly illustrate that complex life cycle we mentioned earlier.
Alternation of generations.
They do. In the kelp life cycle, you have a clear switch between a multicellular diploid form, the sporophyte, that's the huge kelp plant you see, and a tiny multicellular haploid form, the gametophyte. The cycle requires both stages to exist independently.
Before we leave Straminophiles, we have to mention the Umicides, the water molds. They fooled biologists for decades.
They did.
Yeah.
They look and act so much like fungi that taxonomy literally got tricked by evolutionary convergence. Umicides are decomposers and parasites, but unlike fungi, which have chitin cell walls, Umicides use cellulose.
And they can be devastating.
Oh, agricultural terrorists. Phytophthora infestans is the umicide that caused the potato late blight, which triggered the Irish famine.
And a brutal reminder of their power. Okay, next up within SAR, we have the alveolates.
The defining feature here is cellular architecture. They have these flattened sacs called alveoli located just under the plasma membrane. This clade includes three main types. First, dinoflagellates.
The ones that cause the red tides.
Exactly, those famous flagellated mixotrophs. Then you have the epicomplexans. These are almost all specialized animal parasites. They spread via infectious cells called sporozoites that use a complex of organelles at their tip, the Evo complex, to literally drill into a host cell.
And this is the group that includes Plasmodium, the agent of malaria.
Yes, Plasmodium is a master of complexity. It needs two hosts, humans and mosquitoes, to complete its life cycle. And it has this fascinating remnant, a non-photosynthetic plastid called the apicoplast.
Which it got through that secondary endosymbiosis.
Likely from a red alga, yes.
Hmm.
And even though it can't photosynthesize, the apicoplast is vital for its survival, which makes it a major target for drug development.
And the last of the alveolates are the ciliates, like paramecium, using thousands of tiny cilia for movement.
And they're also unique because of their nuclear duality. They have a small micronucleus just for sex, and a large macronucleus that runs all the day-to-day operations. It's a really efficient division of labor.
Okay, the last group in SAR is the rhizarians. These are the amoebas with those distinctive thread-like pseudopodia.
This group is all about beauty and geology. It includes foraminiferans, or forams, which secrete these porous, hardened shells called tests made of calcium carbonate. And their tests are incredibly common fossils.
Which makes them crucial for geology, right?
Absolutely essential. Different species live during specific time periods, so finding specific foram fossils is how scientists date sedimentary rock. They're also vital for tracking past climate change. And their relatives, the radiolarians, create these astonishingly intricate, symmetrical silica skeletons.
Moving on to our third supergroup, Archaeplastida. This one seems more straightforward, representing the direct descendants of that primary endosymbiosis event.
Exactly. This is the line that first kept the cyanobacterium. It includes red algae and green algae, plus all plants. Red algae get their color from a pigment called phycoerythrin.
And that lets them live deeper in the ocean.
It does. It allows them to capture the blue-green light that penetrates deep underwater. Many edible seaweeds, like the nori you use for sushi, come from this group.
And the green algae are our closest protist relatives, sharing so many characteristics with true plants.
They show incredible diversity in how they got complex. Some form colonies, some are truly multicellular, like sea lettuce, and some, like the organism Caulerpa, get big by creating a single, multinucleate supercell without any internal walls. It's just a different way to get big.
Finally, we arrive at the fourth and final supergroup, Uniconta, which includes the protists most closely related to animals and fungi.
Unicontis is split into amoebozoans and opisthocants. The amoebozoans are defined by their lobe or tube-shaped pseudopodia, which they use for moving and feeding. This sets them apart from the thread-like ones we saw in rhizarians.
And this group also contains the famous slime molds.
The slime molds are incredible. Another case of evolutionary convergence with fungi. You have two types. The non-multicellular plasmodial slime molds form this massive, single, brightly colored, multinucleate mass, literally a supercell that can stretch across logs.
And then you have the cellular slime molds, which are more social.
Right. Cellular slime molds like Dictyostelium live as individual amoeba-like cells. But when food runs out, they all aggregate and form a collective slug-like unit that migrates before forming a fruiting body to disperse spores. It's a temporary multicellularity driven by starvation.
And the opisthocants include us, animals, plus fungi, and their protist relatives, which just confirms why that old single-kingdom classification for protists had to be abandoned. They're scattered everywhere.
So we have mapped this stunning complexity in history. Now, why does this catalog of life matter outside of a biology textbook? What are the crucial ecological rules?
They are foundational. First, as symbionts. In mutualistic roles, they're critical. Photosynthetic dinoflagellates feed coral polyps, making coral reefs possible. They also digest wood in the guts of termites. And on the flip side, their parasitic roles are globally devastating.
Absolutely. From the dysentery caused by Entamoeba histolytica to the devastation of Plasmodium and the economic damage from plant pathogens like Phytophthora ramorum, sudden oak death protists pose massive challenges.
But their truly vital role, as we hinted at, is as producers at the very base of the food web.
They are fundamental. Photosynthetic protists and prokaryotes together account for roughly half of the world's photosynthesis. Diatoms and other algae alone are responsible for nearly a third. They underpin all aquatic systems.
And this is where their fragility connects directly to global climate change.
Precisely. Researchers have observed a really worrying decline in marine producer biomass, fewer algae, fewer diatoms in warmer ocean regions since the mid-20th century, and the hypothesis linking it to climate change is compelling.
How does warming temperatures specifically hurt producer growth?
Rising sea surface temperatures create this stable, warm layer of water on the surface, and this layer acts like a barrier, preventing a crucial process called upwelling. Upwelling brings cold, nutrient-rich water from the deep ocean back to the surface, where the protists live. If that warm barrier stops the nutrients from reaching them, the foundational producers starve and their populations decline.
So that feedback loop warming temperatures hindering the ocean's biggest natural carbon sink, that's a monumental concern.
To synthesize what we've explored, protists are these essential, incredibly complex eukaryotes that just don't fit into the animal, plant, or fungus boxes. Their diversity is rooted in the single event that gave us mitochondria and the multiple events that gave us plastids via endosymbiosis.
And this complexity is now classified into the four major supergroups: excavata, the sprawling SAR group, Archaeplastida, and uniconta. They use these specialized structures to live everywhere, from deep oceans to our bloodstreams, and they underpin global ecosystems as producers and recyclers.
And here's a final thought to leave you with. Built on all this complexity, we saw incredibly complex organs like the ocelloid eye and multicellular body plans like kelp evolving completely independently in separate protist lineages.
So, separate from animals and plants, this phenomenon, evolutionary convergence, where similar optimal solutions just arise again and again, it raises a huge question.
It really does.
Does the complexity of life over long time scales naturally push organisms toward a fixed set of best biological designs, regardless of where they started?
It suggests that maybe, just maybe, the environment dictates structure even more than shared ancestry does. A true deep dive for another day.
Thank you for joining us as we explored the most diverse and complicated part of the eukaryotic family tree.